So I want thanks for everyone who's joined. Hopefully, we'll get a few more people and everyone's safe. It's my pleasure to introduce Logan for faculty who haven't seen or might have seen him. Logan joined the department recently as a postdoctoral fellow, and Bob Benedict is his mentor as part of the T32 we have at Emory on vaccinology. So I'm sure it's no surprise that he is very interested in looking at vaccine hesitancy. But he comes with a strong biological base. He's an alum of Emory, finished his PhD from IMP. Is that right, Logan?
Yeah.
Immunology and pathology. So he's truly integrating basic sciences with behavioral sciences for global health. And Logan, looking forward to hearing what you're doing, what your plans are. And again, Logan is very much in the department. We bump into each other. He's located where Carla and or that side of the sixth floor. So once again, thanks for agreeing to do this. And it's a great way for you to meet uh, more faculty and share your interests. All
Yeah.
yours.
Thank you. Thank you. I think I can share my screen, so hopefully this all works.
Yep, it looks good.
All right, cool. And is it the full screen version, or are you guys getting the presenter view?
Yeah, this is
Now,
good.
We're good? Okay. All right. Well, thanks everyone for coming. As I was mentioning to Usha earlier, not the biggest fan of Zoom presentations. I tend to talk with my hands like I just did, and it feels really weird to do it in a room by yourself. So excuse me if I'm a little awkward from time to time. But so today, as Usha had mentioned, I'm here to talk to you about my work from the Vaccinology T32 at Emory. It's mostly a lab-focused T32. However, Bob is one of the mentors on the grant, and I use this as an opportunity to kind of expand my knowledge base and move into the realm of public health. So today, I will be talking to you about a vaccine technology called microarray patches, and specifically how they can improve vaccine delivery and address health equity, but we are unsure if people are going to actually be interested in using them. So here's a brief outline. So we'll go through some vaccine technology and vaccine background, some more specifics on the microarray patch, the implications of this on a global and domestic level. I'll highlight the methods that we use to assess our data, some of the results, and then some of the things we have coming up. So a little bit of a background about myself. I started off getting my bachelor's in biochemistry at the University of Oklahoma. I did some cancer treatment research there, focused on interactions of proteins in cancer cells and their role in various comorbidities. I then did a post-bac as another biochemistry post-bac looking at metabolism in Drosophila. So a lot of fun playing with fruit flies. Not the greatest thing ever. And then finally, I came to Emory and did my PhD in immunology. And this is where my research kind of shifted towards the virology, immunology, vaccine side of things. And interestingly, I did my PhD work over at CDC under Paul Rhoda with a focus on measles virus and measles vaccine and its role in its interactions with B cells. And lastly, here I am now doing a postdoc at Emory, and we're looking at sociodemographic factors of vaccine hesitancy, but specifically with a focus on new vaccine technology. And just moving on from here, if anybody has a question or anything, feel free to interrupt me. Okay. So, kind of expanding on this, as Usha mentioned, I have a pretty lengthy background in biological sciences and laboratory sciences. And luckily, that took me to CDC, which allowed me to gain more exposure to public health than a lot of lab-based scientists would have normally. And I really realized during this time, I really enjoyed doing presentations, giving talks, presenting posters, and just really talking to people about science and trying to, learn how to educate folks on the things that are going on in our field, including those that are colleagues as well as those that are, lay people. And so because of this strong interest, this made me decide I wanted to pursue a career that was more focused on this communication and education side of things. So that's whenever I found the T32, and saw that Bob was one of the mentors on there. And I thought, well, this is a great way to tie in my interests across the board. And, lastly, I come from a community in Oklahoma that has, a lot of underrepresented socio-demographic groups. And, I've seen the impacts of, health inequity and stuff firsthand. So I just, really tie in everything in my life, some of my experiences from when I was younger, a lot of my research experiences, and finally, like my research interests. So this T32 has been a really great opportunity to tie in everything that's important to me. But, let's get to some vaccines. So to start, we'll get everybody on a baseline of some vaccine background here. So importantly to know, the reason we use vaccines is because they prime our immune response to respond more quickly and more effectively to future pathogens that our body encounters. And here's a graphical representation of that. Let me turn on this laser pointer. So we can say that here on this first one, we have our immune response, and this is going to be our initial exposure to whatever the vaccine is or antigen, what have you. And then, so this is the vaccine. And then when we have a secondary encounter, we can see here just that there's a larger response. And if you look at the timeline, it is much quicker. It's about half the time. So that's how vaccines work in the short and simple form of it. Basically, they just, prepare our body to react the second time. And there was a paper recently that I need to read that Bob sent out that really takes this and relates it to other metaphors and analogies and stuff that could this kind of stuff could be really utilized in vaccine communication in the future. So we got to think about how we can translate this kind of information into more of a metaphor. Like the one I always hear is like practicing for a sport, right? Um, and so importantly, these vaccines can prevent a number of potentially fatal diseases. And this it ranges from cancer with the HPV vaccine to measles with the MMR vaccine. And I know everybody knows about polio, so the polio vaccine. So the list goes on and on and on. And obviously, not all of these diseases or whatever have high morbidity rates or mortality rates, but in certain contexts they do. For example, with measles, in the U.S., the mortality rate's fairly low, but if you go into locations that have like global health crises, such as in Haiti when they had the earthquake, the mortality rate went up to like 40%, which is a pretty significant number. So there's a lot of context when it comes to the mortality of these diseases, and that's why we should just attempt to prevent them across the board. And here's an example of that. So we can see, super high measles cases. This is globally, and then whenever the vaccine was invented and implemented, it was invented in the 60s and then implemented globally in the 80s. You can see a very clear drop in the cases. So just kind of a real-world example of the vaccines working. And importantly, there are many people who are immune compromised or they can't receive vaccines for a plethora of reasons. And whenever we reach a certain level of vaccine coverage in our communities, this allows us to have something called herd immunity, which can prevent the spread of viruses to those who cannot receive the vaccine. So if you think about it, when someone's exposed, they could take it and expose somebody else that is not protected. But if we limit the amount of exposures that are possible, we have the ability to protect those who cannot have the vaccine. And here's a graphical representation of that. And then lastly, this can be especially helpful in regions with less healthcare infrastructure. So if we have measles in low middle income countries, they're one of the biggest things that affects the mortality of measles is the ability of the healthcare system to take care of those individuals that see some of the secondary infections that often follow measles. And, if we have something that just completely prevents that from having to happen, that lessens the burden on the healthcare system in addition to, protecting the individuals that don't necessarily have better access to healthcare itself. And so what is a vaccine? So there's a number of them, and I'll walk through some of the common ones here. So we have a live attenuated vaccine. This is a live virus that has been weakened there's a number of methods you can use here. Some examples of this are oral polio, MMR, chickenpox, and flu mist, so flu nasal spray. We also have some subunit or conjugate vaccines, and these are generally made up of a portion of the virus. So this can be part of the virus, like a protein or a membrane, or it could be an entire virus-like particle. So this is a particle that kind of mimics what a virus would look like, but isn't infectious. And so this is Hep B, injected flu, and HPV. Then we also have inactivated pathogens, which is a pathogen that has been rendered non-infectious or dead. This is injected polio vaccine, and then Hep A. We also have vaccines that protect us from toxins that are secreted by whatever pathogen is infecting us. So this is called a toxoid vaccine. And this is most commonly found in tetanus. So the vaccine protects us from tetanus toxoid. And then lastly, our new favorite thing to discuss is mRNA vaccines. This inserts protein coding into mRNA, which then our cells in our body will pick up and translate this into protein from whatever pathogen. And this allows our body to build an immune response to these proteins. And so, as we all know, this is Pfizer, Moderna, COVID-19, and there's many others in development. There's been a lot of research on this for a while, actually. So this is all great and handy, but let's put it all in the context of things. We have a very brief vaccine timeline, and a warning, this is not comprehensive. These are just some time points that I thought were important in discussing this. So please don't shame me for not having the entirety of the vaccine history here. So in 1796, we see our first example of a vaccine with Edward Jenner as he inoculates James Phipps with cowpox. So Edward Jenner had an observation with some milkmaids and saw that those who were interacting with the cows were not getting smallpox, and he noticed that they had some sores on their hands, and they connected this to cowpox. So one second, my dog is chewing on a toy. Hey, go. And so they'd use this to inoculate people and carry on with vaccinating those individuals with the cowpox. In 1853, we have the invention of a syringe, which is what we see now with vaccines. And then another notable one is kind of the discovery of epidemiology, really, and the spread of disease in social settings with the Broad Street Pump from John Snow. There's a book about it called Ghost Map. You should read it. I'm not really much of a reader, and I really enjoyed this book. So if you haven't read that, you should take a look at that. Next, we have the invention of another vaccine technology that is used or was used fairly commonly in the mid-1900s. Not as much today, but it is used in some low middle-income countries. The jet injector. Then we have, all this has happened before germ theory was even fully discovered and materialized, which happened in 1884 with Robert Koch. And then we have the usage of these jet injectors that I mentioned before. I know this is a big jump, but there was mass vaccination using jet injectors. However, because of the way it worked, there was blood spray whenever you would use it in some cases. Oftentimes, they would take the jet injector and just go down a line of people. And this had a lot of contamination concerns. In 1955, we had the inactivated polio vaccine invented. In the 50s and 60s, we have nasal sprays invented. Initially, it was used for various like anti-inflammatory stuff, not necessarily vaccines, but this is when the technology itself was invented. And then we have the invent of the bifurcated needle, which helped a lot with administering smallpox vaccine and leading to the eradication of smallpox. In 1961, we had the invention of the oral polio vaccine, which is the one that was used globally to really help with the eradication of polio itself. And then in 1968, we began measles vaccine distribution. Again, another very important vaccine that's being distributed, and a lot of people are aiming to make measles one of the next viruses that is eradicated. There's a lot of logistic factors that go into that, though. And then we move from regular jet injectors to disposable jet injectors. And this is kind of where they are used now. They use single-use jet injectors that come in a packaging. They're disposable. They don't have this concern about spraying blood or contamination from individual to individual. And then finally, we get to kind of the current state of affairs. And I say finally, even though it was over 30 years ago in 1993, they begin to test mRNA as an immunogenic molecule to see how this could be used in the future. So in 1993, they discovered that mRNA generates an immune response in their hosts. Um, in 2003, we're just tying in some more of these technologies into currently used vaccines. We have the influenza mist was released. And then another technology, which is the one that I will be focusing on for this talk, is the microarray patch. It was patented in 2007, and I know this is there's not a lot of context here, but I'll get into some more background and function on the microarray patch here in a second. And then in 2010, we see our first mRNA preclinical cancer treatment. So again, I want to put this in the frame that this technology that everybody thinks is new and scary or whatever isn't necessarily a new technology. It's been being used for over a decade now. And this is one of the first clinical trials we see it being used in as a treatment. And then we moved to 2017, so 3 years, 4 years before the mRNA vaccines are invented for COVID, and we're already testing mRNA vaccines in 2017. And then focusing back on the microarray patch, there's a MAP Center of Excellence formed focusing on low middle income countries. So we can see how this is moving forward to understanding this technology and its like global implications here. 2020, we have the COVID-19 vaccine trials. And at the same time, these microarray patches are given high priority by global vaccine conglomerates. And then lastly, we currently have an ongoing clinical trial using these microarray patches for MMR. And so this is probably a little confusing. So the reason I put the microarray patches side by side with this mRNA is because we want to look at like the timeline of when these things were invented and when they're integrated into our society and kind of the reaction that people have to them, because the current state of vaccine hesitancy has had a lot of impact from the COVID-19 vaccine release. This new technology that people don't have any kind of understanding of, specifically, in the general population, they don't understand how it works. They don't understand what mRNA means. And so this, there's a lot of fear mongering going on around it. And so this leads to, some hesitancy focused on the mRNA technology itself. And so with that, and we have this new technology, the microarray patch or the MAP, coming out and going through clinical tests kind of in like a similar time span of over 20, 30 years. They're starting to test it, put it in clinical trials, and hopefully in the near future use it for vaccines. The whole impetus of this is to take these lessons that we learned from the mRNA hesitancy and the technology surrounding it and apply it to the new vaccine technology that's coming out, especially since these technologies can have such a strong impact on health equity problems, distribution of vaccine, et cetera. We want to make sure we get ahead of this hesitancy that is associated with the technology. So that way there's more acceptance, more positive perceptions, et cetera. So kind of moving on to the vaccine hesitancy, this has been going on for a very long time. This is a comic or a cartoon that was released back whenever they were doing cowpox vaccination. And as you can see, people thought that if you got the cowpox vaccination, you would develop, morbid cow-like features. You would grow cows out of your arms, et cetera. So this was a very real thing. People thought that cowpox vaccine or inoculation would turn you into a cow, simply. So this idea of vaccine hesitancy isn't new either, and it's been taking us over a century at this point to kind of understand it and get past it. And so generally, whenever we're talking about vaccine hesitancy, there's a few models that you can do to model how it works and how it like translates into like the general population. But there's a newer model that's been adapted called the 5C model of vaccine hesitancy, and it focuses on 5Cs that really inform and drive vaccine hesitancy. And so the first one is confidence. and that's pretty self-explanatory. This is the trust and the effectiveness and the safety of the vaccines. The second one is calculation, which is something that is a strong highlight in education and communication materials, is giving people the ability to weigh the risks and benefits of vaccines. Most people aren't trained in like risk benefit analysis and stuff, so making that a more publicly available method of thinking can help address vaccine hesitancy. Convenience. So this can range from distance to a healthcare provider, financial convenience, or just psychological or physical barriers to access what they need. Complacency. Some people don't. This goes back to kind of weighing the risks. Some people don't think that vaccines are important because they're not worried about what could there's other things that are going on that are more of a priority to them than getting vaccinated. And then lastly, we have collective responsibility, which is the willingness to protect others through vaccination. So this is, a social concept of if I get vaccinated, then I'm protecting these other individuals, right? And so kind of zooming back into the mRNA and new technology side of things, this is a breakdown of a study that was published in Nature Medicine. So we can highlight that there's, strongly agree, et cetera, all the way down to strongly disagree. You can see that there's a range and a range of reasons that people are hesitant about the vaccine, and also a range of feelings about the vaccine. So, here in this first one, we'll just highlight that many people are hesitant. I say hesitant with quotes about the mRNA vaccine because they don't know the long-term effects of the vaccine, and this is more than 50% of people, right? So this is again something that's like, if we take this concept and educate people on how mRNA technology works and why this isn't something they should be concerned about, we can theoretically shrink this number. And the reason I keep focusing on this idea of new technology is because if we take a bigger picture look at vaccines as a whole, there's a couple studies that have been put out by Pew Research Center over the past couple years. This first one is really one that I want to highlight, is that this hesitancy isn't necessarily a broad stroke across all vaccines. While we do see a dip in vaccine uptake across many vaccines, when you look at like the perceptions of the vaccines, if we focus here on this left set of bars, you can see that whenever it comes to like risk analysis, people don't think that the COVID-19 vaccine is worth the risk. But if you compare that to a childhood vaccine, such as MMR, that has been around for a while, isn't this like new technology? There's a huge shift in the perceptions of these. And so there's a number of things that could be fueling this. I think, Bob and I have talked at length about what could be, fueling these kind of feelings, how we can like address them and stuff. And I think, sorry, one of the big things that we've observed over the past, few years is that there's been this large shift in the trust in scientists and public and healthcare officials. And so you can see that there's this big shift down. It was like a 10% shift down over the past 5 years of trust in scientists. So there's a trend downwards in the trust of those that generate these healthcare things and are the ones in charge of researching it. And I think that is an issue propagated by something that I've like anecdotally also witnessed, is that a lot of times there's these researchers and scientists that need to learn and hone their ability to communicate their scientific findings to others. And so this is reflected in the general populace where a lot of people respect the intelligence of these scientists. However, they say that the majority of people do not feel like they're good at communicators. So that's why when we take these new technologies, be it vaccines or some other medical treatment, we need to make sure we have people in a position that can communicate these technologies well to, address a lot of these concerns. And so that's kind of where people like us come in. We do the social and behavioral side of things to really understand the perceptions of these, and then we communicate with the scientists and stuff and become kind of like a liaison to communicate these findings to people in a way that is effective to them and that will address a lot of their concerns. And so, as we all know, a lot of these concerns and stuff can all vary based on, sociodemographic group. So we can really hone in into each of these groups, find out what their concerns are. So let's say, like in a Hispanic population, they might have a different concern than those who identify as like non-Hispanic Black. So we really need to consider these things when we're making communication and education materials for these new technologies. And so I know this is a lot of background, so I want to make sure that everybody is on the same page. So we move into this new technology of this, these vaccine technologies, and there's a number of them that are non-needle based. And, one of the big factors of vaccine hesitancy that most people probably don't think about is needle fear. There's a roughly 24% of people have some sort of needle fear, which can impact vaccine hesitancy. But there are also other implications in vaccine technology, such as immune response, ease of administration, et cetera, that can play a role in why people administer a vaccine a certain way. So here's just a handful of vaccine technologies that are utilized now, all the way from microarray patch to nasal administration, a lot of the things I highlighted in that timeline. But as you could probably have guessed, I want to focus in on this microarray patch, and specifically focus on the idea that before we can effectively roll out this technology, we need to first understand the opinions of them and how this can vary from demographic group to demographic group, and really hone in our communication strategies for this. So to start off with that, we have this in our survey that we did. I'll get into the specifics of the survey. We're getting close to the results, don't worry. We did an acceptance of all these devices, and as you can see, there's a very clear trend, and shockingly, needle was, so if we look at the black, these are people that would not recommend, this is the percentage of people that would not recommend this vaccine methodology for anybody, themselves or others. And as you can see, needle was ironically the most accepted one, and this declines over, several, over all the vaccine types. Inhaled being the least accepted, with skin patch being right here in the middle. So you can see that there's not a whole lot of acceptance of this just from like a first look at this device. So that really means we have a lot of work to do when implementing this. And so how do these vaccines work? So again, these can avoid needle fear. We moved away from the term microneedle array patch because we don't want to use the word needle. But basically, it's a small Band-Aid, as you can see here, with a bunch of tiny little spikes. These spikes can be anywhere from 100 to 1,000 micrometers long. So not very big, with a typical, subcutaneous needle being 25 to 38 millimeters. And this is about 31 times the size of these little needles. So if you shrink it by 30, that's the size of these needles. And the way they work is they are put on like a Band-Aid, and this allows, insertion of these spikes into the skin, and this allows for a release of whatever's in the vaccine. This can also be used for drugs. And based on the formulation of the needle, you can have either a slow or fast release of whatever content is in there. And so some of the other things they're using it for is like birth control. They have extended release birth control in these patches. The needles will, when they pull the backing off of the Band-Aid, the needles will stay in your arm and continue to release whatever's in them. And importantly about the way this works is when the skin is penetrated by these spikes, is that this can be stimulating for the immune system, kind of like when you get a scratch or a cut. This recruits immune cells, and luckily there is a high density of immune cells in your skin that can strengthen the immune response. So you have this quote unquote wound in your skin that attracts immune cells, coupled with the immune response to the vaccine itself. So it's a really good microenvironment to generate a very strong immune response. And there's a number of papers that focus on the immunology of the skin and how it's a good place to formulate an immune response. And the best part about this, just like the mRNA vaccines, theoretically, this methodology can be used for any kind of vaccine, specifically ones that will generate an immune response in the skin. I know there's work on, obviously there's MMR, there's work on HPV vaccine, there's some flu ones, there's a bunch of them ongoing right now. But what does this look like at like a global scale, and how it addresses health equity? Well, there's a number of things that impact our ability to, distribute needle and syringe vaccines to, low middle income countries, regions here that are impacted by like distance to healthcare and stuff. So I have a couple highlights here. So I think the biggest one that I think is probably going to be the most important moving forward is that microarray patches, and several studies have shown a large amount of thermostability. So needle syringe, you need a cold chain a lot of the time, and sometimes this can be very cold, -20 C. all the way up to like refrigerated temperature, and the stability of vaccines in each of these contexts depends on the vaccine. And so this means that we have to consider the logistics of transferring a vaccine from point A to point B and all the storage conditions we need between point A and point B. And the patch, what it does is it addresses this in a lot of contexts. It can address this storage. In one of their studies they did with MMR, is they saw that for up to 3 months, this patch can be stable at 40 degrees Celsius and still elicit an immune response that's strong enough to obtain immunity. So there's the potential for this patch to hopefully mostly address these logistic concerns around cold chain storage. They're cheaper. There's and in theory, these don't need a trained healthcare worker. They're still working on the specifics of like location and guidance on that. This they have administration things where it's like it kind of just you press it and it pops it in instead of like having to push it on like a Band-Aid. But there's the potential for self-administration. Um, you have to go to a trained healthcare worker if you're living in a rural area. So, this implicates distance and difficulty of travel. And with the size of these, the thermal stability, et cetera, there's the potential for delivery, which is one of the things we question about in our survey. And this kind of is highlighted by the size and packaging of it, the size and all that stuff. And lastly, needle fears, which I highlighted earlier. So again, we have some ongoing research. A lot of the formulation and engineering of this has been going on at Georgia Tech. And there's a lot of clinical trials going on. Specifically, they're starting a phase 3 clinical trial of MMR in the Gambia. What they saw in phase 2 is it has similar immunostimulation and more seroconversion. And here's where we come in. So the current assessments of perceptions and acceptance of these have been done in urban settings or in clinical trials. And the reason what we're doing is different is because clinical trial participants are probably more receptive to new vaccine technology anyway, because they're willingly signing up for a clinical trial. Urban populations are not generally fully representative of, the population overall. And while urban populations could benefit, as I highlighted in the last slide, a lot of benefits could be given to underserved communities. So we need to have a broader understanding of how people feel about this, because that'll allow us to address the gaps in equity, tailor logistics to certain groups and communications to certain groups, and address group-specific hesitancy, and find ways to disseminate information and address misinformation effectively in certain communities. So this is kind of the entire background of my life and how we get to this point we're at now. And so what we wanted to do is assess attitudes toward these vaccine delivery devices. We did all of them that I showed previously, but I want to focus on the microarray patch for this talk. And we wanted to assess the patterns of these attitudes compared to non-needle-based methods. And then we looked across different sociodemographic groups to see how these patterns change. So going into methods. So the survey was designed by me, Bob, Mark Krasnitz at Georgia Tech, who is one of the PIs for the labs that engineer the patches. Kira from our team, and then Erica Spence, who was one of our grad students. We distributed it through Qualtrics, and we specifically wanted to correlate to census distributions for Georgia specifically, so that way we're getting a more representative population, and we focus on race, gender, and urbanization rates in Georgia. And then we use SAS for data analysis. We reached out to 698 individuals. We screened out 271 for various reasons. This includes intention checks, non-Georgia residents, English proficiency, etc. And we ended up recording 427 responses in our survey. And we broke down some of the sociodemographic characteristics into buckets that make sense for analysis. So for age, we thought, access to education and, experience with new vaccine technology and stuff over the generations was an important way to break it down. So we have our age broken down into generational groups according to Pew Research Center's guidelines. Race was self-identified, and we have non-Hispanic Black, non-Hispanic White, and Hispanic. And then we have the other category, and I would love to not have to have this other category and have the granularity of each of these groups specifically, but sadly the N was for this group was not very high. In an ideal world, I would have a separate one for everyone. And then for education, we have high school or less, some post-high school, be it trade school, associate degree, or just, some of that, bachelor's degree, and then higher than bachelor's degree, which includes some graduate education, master's, and doctoral. And we looked at vaccine hesitancy and trust of new medical developments based on Likert scales, which are, not at all to vary, right, on both sides. And then we had that graph I showed you earlier that showed the people's acceptance of the vaccines themselves, and this was whether or not they would recommend them to sell for themselves, for their spouse, or others. And then we move specifically into the acceptance of the microarray patches. I'm just going to say MAP from here on out. The perception of specific MAP characteristics, the barriers to vaccination that people experience, and lastly, the the locations that people receive their vaccines. And so just as transparency, anybody that answered, prefer not to answer, we removed this from the analysis. Not that we just removed their answer from that question, not the individual themselves. This is just a very brief window of all of the variables we assessed in this survey. So if anybody's interested in vaccine hesitancy, and any other variables that I have, I would be happy to talk to you because there is so much data in this data set, and I am just one person. So feel free to reach out. So just for some sake of presentation, here's some abbreviations I'm going to use. So PR is prevalence ratio, APR is adjusted prevalence ratio, and then these non-Hispanic white, non-Hispanic black, confidence interval, and ref refers to the referent group whenever I'm talking about the analysis. So to start, vaccine hesitancy. So the questions at the top, in general, how would you rate your hesitancy towards receiving a vaccine? And a surprisingly high number of people registered as hesitant or non-vaccinating at 52.6%. Other literature states that the general range for the United States is 23.5%, with a range from 11.7 to 40.2 based on what state you're in. And this is a pretty big sample size they had. So my survey, the Georgia responses were fairly high. However, we did notice some trends based on what sociodemographic group we stratified by. So groups that were generally less hesitant, looking at multivariate analysis, or groups that identified as Hispanic compared to non-Hispanic whites, they were less likely to be hesitant. But there was much more groups that were more likely to be hesitant compared to the referent groups. So basically, anybody under the age of 59 was more likely to be hesitant, which is kind of a shocking concern moving forward because these are all the people that are going to be, around making healthcare decisions, getting their children vaccinated, et cetera. So these are important targets for, addressing this vaccine hesitancy. And the younger they are, the worse it was. So something to pay attention to. We had an income group when compared to low income groups of this 100 to 150,000. Not sure necessarily what that means moving forward, but something to focus in on. And then obviously we assessed the trust in medical developments, and this is kind of a hypothesis testing here, is that the people who have low trust in new medical developments are probably going to be vaccine hesitant, and this is kind of what this confirms here. And so specifically on the trust in new medical developments, surprisingly, even though there was this high amount of vaccine hesitancy, there wasn't that big of an amount of people that have low trust in new medical developments, only 14.5%. So the more trusting groups, there's not a whole lot of, there wasn't a lot of multivariate analysis that led to significant results here, but the more trusting groups that were from this analysis were non-Hispanic blacks and low vaccine hesitance individuals, which again kind of makes sense. So, these are groups that we need to consider whenever we're communicating. They may not need as much focus, or they'll be easy to convince with new medical technologies, et cetera. But, as many people I've talked to over the past year and a half when focusing on this research, it really boils down to how much do you know, have you heard of these things, et cetera, because if you have a good baseline going in, you're more likely to, be open to information about what have you. And so what we did here was we asked which of these vaccination methods have you heard about, and then select all that apply. And so there's a few that I want to focus on here. The first being this none category. So we listed out all these vaccination methods as they're shown here in the graph. And still around 8% of people that took the survey said they had heard of none of these vaccination methods, which is, not a whole lot of people, but it's still a striking number that could lead to concern. Then we look at this needle syringe one. And so again, kind of a shocking number. Over 20% of people said they had never or did not select that they had ever heard of needle or syringe, which I'll kind of go into why that might be here in a second, but that's still a shocking number of people for something that's so common that they've never heard of it. And then lastly, my focus of the talk is the skin patch. So only about 20% of people have heard of it. And it's pretty commonplace in like the scientific realm right now. It's like it has a lot of implications, et cetera. So we really need to make sure we're disseminating information to these people ahead of time, so that way whenever this technology does slowly start to get implemented into our existing infrastructure, they're aware of it and kind of understand it. Yeah, I'll come back to this momentarily. So we're going to analyze each of these groups individually and look at the bivariate multivariate analysis. So we noticed that those who were the most likely to know any of the devices that were listed were the low vaccine hesitance group, which makes sense. They're aware of vaccines. But the ones that are less likely to know any of the devices are this specific age range right here. So I believe this would be Gen X. They were pretty unlikely to know any of the devices compared to the other groups. But again, this is a low sample size, so just take some of these with a grain of salt. Various insurance characteristics. So those on Medicaid, those using TRICARE, which is more of a military-based insurance, and then those who are kind of unsure of their insurance plan. So these are all underrepresented groups that we need to focus on, that are generally focused on in research now. So this kind of drives home the point that these consistently underrepresented groups need to have focus here as well. And then those that have low trust in developments, which, maybe because their knowledge of the field isn't as high, they have this low trust, right? So it all kind of comes back to the same idea. So then moving on to this needle syringe group, 77.99% of people had heard of a needle syringe, but there's still that 22% of people that have never heard of it, which is shocking and concerning. So the ones that were less likely to know the needle and syringe was this Medicaid group here, which is 37% of the people that were on Medicaid, according to the survey, did not know what it was. So these are people that, are underinsured. They don't have a lot of times don't have the income necessary to, receive medical treatment and all this other stuff. And they don't know one of the, more basic opportunities they have to protect themselves. So again, this is an underrepresented population that could be benefiting from, technology, from vaccines, or just vaccines in general, that we need to focus in on. But specifically, what can we learn from the context of this survey when it comes to this? So we said, have you heard of needle/syringe vaccines? This may not be the word that is communicated from a healthcare practitioner. So this kind of goes back to the ability of scientists and researchers to communicate. Do we need to change the word from needle syringe to just shot, or, as has been common over the past 5 years, just the jab or something like that? If we change the language in our survey and analysis tools, does this change this number? So this really kind of drives home the point that we need to focus in on how we communicate things to people and test messages and really make sure that our message is getting across. And then focusing back in on my technology, only 80 people, or 18%, knew of this. So less likely to know were non-Hispanic Blacks, the same Gen X age group, those who had an education of high school or less, and these were reference group, sorry, to this was the some post high school, and we did that based on the N of these groups. And those who had a bachelor's degree, and then finally, those who had a low trust in medical developments. So really, what does this mean? From a survey standpoint, we kind of just get a quick image of what's going on. We have, a lot of descriptive epidemiology that can happen here, where we can just see percentages and stuff like that, and drivers of these these questions. However, to really get why do these groups have, less knowledge of this, or how do they feel about these things, we really need to move into some qualitative work, which will then lead to, message testing. We can design messages based on this, and then we can generate educational materials that effectively educate these groups or communicate to them, so that way there is a broader scope of knowledge in these groups moving forward. So the survey is really just like a baseline to form, bigger picture things. And highlighting these groups can allow us to know where we need to focus our efforts. And then we move specifically into non-needle acceptance. So we have, the needle syringe vaccine, but now we want to see how people feel about any kind of vaccine that is non-needle based. So 125 people, so roughly, I should have put the percent on here, roughly a quarter of them were more likely to accept. And the most accepting group here, and the only one that was significant based on multivariate analysis, was those who identify as Hispanic, which is consistent with literature. Generally, there's a study in Alabama that showed that, Hispanic populations are generally more willing to accept vaccines, and they have been pretty good about uptake over the past few years. Those were that less likely to accept. And this is the one I'm sad to see moving forward, especially with the intent of my research and, the goals of this type of the goals and potentials for this vaccine technology is the rural group was less likely to accept. And again, non-Medicaid government benefits. So again, another underrepresented population. And then those with low and medium trust compared to high trust. So really want to make sure we pay attention to these rural groups and these, differentially insured groups when designing communication and education tools. And then, interestingly, those who didn't really care how they received their vaccine, is it via needle or non-needle? Again, this goes back to the Hispanic group and those with low vaccine hesitance. So this is, again, consistent with the literature. And just overall, what we can learn from this is that these groups are willing to get vaccinated. And so, obviously we don't know until we do it, but it seems like, we can just reach out to these groups and they'll be relatively easy to talk to, and, they'll be more willing to take, vaccines as a whole in this context. Some of the breakdown of the reasonings people said they were or were not willing to have a non-needle based method. So yes, I'll just highlight the top three here, but here's all of them. So for the yes, there's no pain, there's no needle, and interestingly, they're interested in new technology. So, that shows that they understand that there is new technology on the way in the field of health and that they're interested in it. And then no, as expected, they just don't trust new vaccines, which again, how much of that can we, snip based on, effective communication and education materials? They're comfortable with current vaccination methods, which is, important to consider. It's like, why, if it's not broke, don't fix it, right? And then the next one I think is important. The next two really, they need more info on the technology or on the vaccine, which shows that these people are hesitant, but only because they're not fully informed of what's going on. So maybe if we give them the materials they need to come to a point where they're comfortable with this, just based on communication materials, we could have them move over to the yes column. So this is all kind of just driving what we should focus on and how we should, generate our outreach for vaccines. And then lastly, we'll kind of hone in on this vaccine recommendation here. So this is the images we showed. So based on, would you recommend this for anybody? So this is the needle syringe one, and then this is the patch one. So just kind of generally what it looks like. And I want to focus on these two columns where here only 20, which I kind of highlighted earlier, 22.4% said they wouldn't recommend needle or syringe to anyone. And then 42.7% said they wouldn't recommend the skin patch to anyone. So there's still a very large gap between what our baseline is and what the new technology is. But again, this is just at like a first glance, first interaction with this technology. How does this make you feel? Would you recommend it to somebody based on this image and the short description? And the short description for this was similar to a Band-Aid. So at first glance, people are pretty hesitant to, do this, even compared to like current methods. So what we can learn from this is, We can think of more intentionally about the image provided. Again, going back to does the way we word things impact people's perception? So does the image we provide impact it? Think about how we describe it. So similar to a Band-Aid, do we need more or less detail? Since people wanted more information on the vaccine, do we need to include, more specifics about the immunology of it? Another thing to consider, does the order of the images impact people's perceptions? If we move needle syringe to the top versus the bottom or in the middle, does that change the answers? There's a lot of nuance here trying to see how people's perceptions are affected by small, minute details that we can consider when we're designing messaging. Yeah. And so getting into the end here, I'll try to wrap up pretty soon. So there's a lot of characteristics about the map that we're looking at specifics for. So this is application time, location, etc. This is just some descriptive statistics of this. I'm working on the bivariate, multivariate analysis of this for now. One second. So here, looking at the 5 minute, again, this is most people are just as likely to accept it with, some being yes, they'll accept it, with some no. These numbers all shift down a little bit when you increase the time of application, and then you get more no's. And then kind of just a brief picture of like, would you take one that was in the mouth? Which, was a very interesting question. Mark really wanted it on, Dr. Prousnitz over at Tech really wanted this in there because they're looking at oral stimulation for vaccines. People weren't really a fan of this idea, but some were willing to do it. So, but again, working on the the sociodemographic characteristics, what drives these opinions for these, and so going into some of the health equity stuff, I looked at to see if people would be willing to have this delivered to them, or if it was available for just pickup from pharmacy or from their healthcare provider, where they don't have to go in and have an appointment, etc. Would they be likely to accept it? Every answer except for equally likely was pretty much the same, all around the 10 to 15%. But it seems like, it wasn't that appealing to a lot of people. So we have people who are much less likely, and equally likely to vaccinate being the majority of it. There are some people that they would be more likely to vaccinate, but not a whole lot. And kind of further, we wanted to look at the effect of urbanization on this, because we want to, this could be particularly impactful for rural populations. And we can see here that the rural population and suburban population compared to urban were, more likely to select less likely to be vaccinated if it was offered through this. Yeah. And so the other thing we talked about was the lack of need for a healthcare provider potentially. And so it actually seems like if it was offered for themselves to apply or the provider to apply, did impact how they felt. So if they were much more likely to, so there's a range of answers here. There was a higher rate of much less likely for self-application, and a higher rate of much more likely for provider application. So, having the provider apply it versus whether or not they could have themselves apply it. And this range, there was some qualitative answers that were, oh, I don't trust myself to do it. So if there was like proper training, that they would be more likely to, etc. So there is a difference between whether or not we have have them apply the patch themselves, or if they had a provider apply it. So again, just more information to focus on when we, try to roll these out. Are people going to be more willing to accept it if they can do it themselves versus if they have their provider do it? Because even if the provider does it, it still addresses like the pain and somebody had mentioned in the qualitative answers, oh, it seems faster, stuff like that. And then last couple things, we try to plan to relate this to where people receive their vaccines. So are people who are receiving from a certain location more likely to be willing to accept these? So most people get them from their primary care provider, but we have a couple other higher ones that they get them from their public health clinic and pharmacy. So pick up from a pharmacy or from a public health clinic, could be something that is beneficial. And then lastly, we have plan to relate to the vaccine barriers. I want to highlight a few of these. So we have, a pretty high rate of people that have no vaccine barriers, which is good to see. Um, these patches can address time because if you can just receive them or pick them up, you don't have to sit around for an appointment or anything. So that gives you more time to do what you need to do. Um, Money, again, these are cheaper. We also, this shows that we need to focus on communicating existing programs like the Vaccines for Children programs. And, most insurances will cover vaccines and stuff like that. Really make sure people are in the know of that. And then going back to really just making sure people know about these things. Do we communicate effectively? Do we educate effectively? And do we, really drive home making this knowledge about these devices and the technology applicable to everybody? And then, of course, vaccine trust, which is kind of what we all know. So really, we got to continue working on knocking down this number. And so, really, to conclude, there's a number of socio-demographic factors that affect vaccine hesitancy, that also affect perceptions of non-needle-based devices. And there's a lot of hesitancy present at the first look at several factors associated with MAP, being location, application time, whether or not people would use it, etc. And so what we can take away from all of this is that there are specific subgroups within the population that we really need to focus in on and make sure when we're designing our messaging and rolling out this technology that we make sure that we address concerns within these groups to really make sure that they understand and we're communicating effectively, which I know I'm preaching to the choir here on this, but it's really just driving home the point that we need to, make sure we really refine our messaging and stuff to make these acceptable and appealing to everyone and not just like look at a broad sweeping brush here. So what we want to do is finish the socio-demographic drivers for these, move into some qualitative interviews, which hopefully recruitment has started for now. Expand both of these, both quantitative and qualitative studies to roll in other underrepresented groups, such as or American Indian, Alaska Native, etc. Expand to other states and regions, because again, this could vary if we're not in Georgia, and then eventually lead to message testing. And then this kind of leads into my next line here. Some other projects I'm working on is, doing some vaccine center policy stuff, more map work, looking at quality assurance stuff from like a manufacturing perspective, looking at, publicly available data with NIS. And, I go back to like the comment I made on my variables where I wish I could have every racial group explicitly out there, really focusing on the granularity of data and how that can be important, especially in these underrepresented groups. Moving towards the qualitative interviews in these populations, specifically in rural populations, helping Bob out with his research. And I also do some minor research on educational tools for undergraduates in biology programs. Moving forward from here, what we want to do is take all of this we've learned here. I'm applying for a KO1 now, so if anybody wants to chat and has some advice or wants to be involved, let me know. And I just like to thank Bob, the rest of our team, our old grad student Erica, and then everybody involved on the Vaccinology T32, Ben, Walt, Nadine, Diane, and then, NIH, obviously, and then Mark over at Tech. And I'm sorry that took a long time. Just wanted to make sure everybody had a good background, but happy to take some questions. I'll stick around and talk longer if people have questions. That'll be after one. Thanks.
Yes. Thank you, Logan. This was a very nice overview, and this is such an important topic. And I know there is a lot of interest right now in I call them a needle. I guess you call them microarrays. Perhaps it's better to avoid the word needle altogether. Yeah. Recently, Barda had made a huge commitment to 4 different manufacturers to make the patch. So that's definitely and speaking of like the nomenclature, I call them microneedle, you call them microarray, but I think your labeling is better, because I do remember when we did the trial at Emory using microneedle patch with influenza vaccine, we were telling people, well, it's not a needle. And it's like, what is it? And it's like, it's like 100 microneedles. So people were like just worried about the numbers of needles they're going to get. I mean, they're tiny and they're velcro-like, as you know. Had anybody kind of raised this question, or the fact that you use the array term completely bypassed this possibility of being needle phobic?
Yeah. And so, since we did like a cross-sectional survey, there wasn't a whole lot of room for people to kind of bring up concerns. But whenever, for the questions that did have the ability for them to, type in whatever they want, nobody really talked about the needle aspect of it. So I think rewording it, I mean, like you said, kind of really drives home the point that we need to work on how we talk about it because, you know, it didn't get brought up at all. So when we move forward with the qualitative stuff, hopefully we get some more information on that because my goal is to, have slides along with the interview so that way we can like walk people through what a microarray patch is. And so maybe whenever we kind of hone into that level, they might have questions. So, all right. Well, I'll go ahead.
I was just gonna say, so I apologize for not being able to get on to introduce you, Logan. I was having some tech issues this morning, but just for everyone on the webinar or on the presentation, I think this, the work that Logan's done really speaks to his commitment to a lot of this. When he came into the T32, this type of kind of public health biostatistics type of analyses was really outside of his main focus area, and seeing how quickly he's taken on to this and been able to take this and run with it, and even take these initial findings and use it to develop a K award application, I think speaks volumes. And I think I'm just saying that to say that as there's potential opportunities for collaboration across the department, if anybody has any interests in kind of these novel devices or new ways of thinking about things, please reach out to Logan and to myself, because I think that it'd be a great opportunities to try to find new ways to integrate them in across the department.
Yeah, and like I said in my earlier on, there's so much data that I haven't had a chance to look at, specifically on like, sources of information, trust, et cetera. So if anybody's interested in that, I would love to work with people on that. All right. Well, thank you, everybody. Wish this could have been in person. Would have been fun. But I sit on the sixth floor and the our Randall Rollins. I can't remember the order of the words. The RRR building. So if you want to come up and chat, just swing by my cubicle when it's not snowing outside.
Thank you, Logan,
Yeah.
for your mentorship.
Thanks, everyone.